will only be important, okay, important topics that we will only discuss with regards to your picornaviruses, but not all the groups of your picornaviruses will be discussed today. Okay, so what are the properties of your picornaviruses? So one of the properties of your picornaviruses, this is the structure. Okay, this is the structure of picornaviruses. So these are RNA linear, okay, linear single-stranded RNA, linear single-stranded RNA, and the shape is usually okay, icosahedral, okay, icosahedral. And each of these icosahedral has a pentamer, okay, pentamer. Okay, so there is usually pentamer here, one pentamer here, one pentamer here, one pentamer. It looks like a tree, but it has a 5 sides, pentamer. So all around, there are 12 pentamers here. Okay, so later na po. Hello, Gemma. Ah, wala nga binanggit si Dr. Trina. Baka may kilala ka, pulmon na o kahit si Dr. Aspiras na lang, siya naman atang sanay sa COVID eh. Oo. Oo, wala siyang binanggit. Tinanong ko kung sino doktor. Sabi niya, wala eh. Kahit sino na lang po. Oo. Sino ang gusto? Sino ang mas okay? Sige, si Dr. Palaboy. Sige. Dr. Palaboy. Wala naman kasi siyang sinabi kahit sino. Sabi naman ni Dr. kung sino ang pando available. Okay, thank you, Jen. Okay. Sorry po. Okay, so it's an icosahedral. So one pentamer, there are around 12, okay, 12 pentamers. Okay, there are around 12 pentamers yan. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Nasa likod yung iba. And each, okay, 12 pentamers, okay, so each, di ba 12, there are 12 pentamers, okay, and you times 5 times 12, so there's around 60, okay, 60 subunits. 60 subunits, okay, in a picornaviruses. Now, this is the pentamer. This 1, 2, 3, 4, 5. So it is a 5, okay, 5 shape, a 5-sided, 5-sided. Now, each of these, okay, pentamer, okay, has 4 polypeptide, okay, has 4 polypeptide. So we have VP1, VP2, VP3. The VP4 is internal, okay, internally located. Nasa loob siya, the VP4, so that you cannot see here the VP4. So this VP1, VP2, VP3 is the this is the capsid. It's the capsid. Okay, so that's the capsid, and that's the one, okay, covering the RNA viruses. So these are the groups of your enteroviruses. We have the polio. We have here the poliovirus. We have the entero A71. We have the entero D68. We have the rhinovirus C15A. We have the hepatitis A, and we have the IK viruses. All of these are members of your enteroviruses. Okay, so this is, as I've said, is an icosahedral. So it contains 60 subunits. So 12 times 5, okay, it's a 60 subunit. And again, this is a single-stranded linear RNA, a single-stranded linear RNA, and it does not contain any envelope. Okay, unlike your herpes virus, it has an envelope. Okay, the envelope galing yan sa host membrane. Okay, they derive it from the host membrane. Okay, so sa herpes virus, there is an envelope, while in picornaviruses, there is no envelope, so an envelope. Okay, so there are 4 major polypeptides. So we have VP1, VP2, VP3, and VP4. Okay, so the VP1 and the VP3 are the major antibody binding site. So it is where the antibody binds, okay, to the VP1 and VP3, while the VP4 is an internal, okay, internal protein. Now there is an genome, okay, so we have the genome link protein, okay, or otherwise known as a VPG, okay, so VPG. So where does replication happen? Okay, the replication usually happens in the cytoplasm. Okay, cytoplasm. Unlike your herpes virus, it usually happens in the nucleus. Okay, but here it happens in the cytoplasm. Now, what are the important outstanding characteristic of your picornaviruses? Okay, so they are made up of mainly enterovirus and rhino. Okay, that infect the human and the lower animals, causing various illnesses. Okay, so the picornavirus causing various illnesses. Ay, Jeff, sandali na po. Sorry for the distraction. Okay, so the clinical symptoms ranging from poliomyelitis to aseptic meningitis to the common cold, urinoviruses. Okay, so it's a wide diseases caused by these picornaviruses. So this is a structure and composition of the enterovirus and rhinovirus. So this is the pentamer side, 1, 2, 3, 4, 5. So there are around 12 pentamers. As I've said, it's a 60 subunits. So we have the VP1 here with the VP1, okay, here, and the VP3 is where the antibody bind. Okay, where the antibodies bind. Okay, so the VP4 is internally located, and this is the VPG, which is the viral genome. Okay, the viral genome, which is the RNA. Okay, the single-stranded RNA. Okay, so it's icosahedral single-stranded positive sense RNA. Okay? So kung positive siya, it has to be converted into negative RNA for them to replicate muna. So from positive to negative for replication to happen. If it's positive, it cannot replicate. It has to be converted first into a negative strand RNA for it to replicate. So later on, we will discuss that. So this is the canyon. You have here the canyon part, the depression between the VP1 and the VP3. Okay, so this is what I'm telling you. This is the pentamer. Okay, so it's a five-fold vertex, and there is a canyon. Okay, canyon. This is the VP1, for example, the VP3. It is where your antibody bind. Okay, antibody binds. Okay, so remember the shape of the antibody is a Y shape. This is the part of the antibody, FAB, where it binds to the VP1, between the VP1 and VP3, which is in the canyon. Okay, so there's a prominent cleft we known as your canyon around each pentameric vertex on the surface of the virus particle. As you can see, the canyon is narrow. Okay, it's narrow, such that the antibody cannot fully penetrate deeper. Okay, because there is a narrowing on the canyon, so cannot fully penetrate. Okay, so there is a poor lock-in relationship. So the receptor binding site used to attach the virion to the host cell is thought to be located near the floor of the canyon, near the floor of the canyon. So the location will probably protect the crucial cell attachment site from structural variation influenced by antibody selection, this one, antibody selection in the host, because the canyon is too narrow to permit deep penetration of the antibody molecules. So dapat, if the antibody binds to the receptor, it should be fully, okay, fully bound para hindi siya ma-detach. So it cannot be detached. But here, it's easily detached because it's not fully bound. Okay, so this is the location of the VP1. Okay, VP1 here. Okay, so this is the canyon. This is the pore of the canyon wherein the antibody cannot fully penetrate deep inside the canyon. Okay, so this is the structure of the capsid. Okay, the VP1, the VP2, the VP3, and the VP4, showing you the structure of the protein. Okay, now what are the differences between the enteroviruses and the rhinoviruses? Okay, when we speak of enteroviruses, okay, so they are stable at acid pH. So meaning when you ingest, okay, when you have taken a contaminated water containing enteroviruses, it can pass through the stomach Okay, so it will not die because it's stable at acidic pH. Okay, it can survive at least 1 to 3 hours. Now, the entero and the rhino are stabilized by magnesium chloride, okay, against thermal inactivation. Okay, so when the temperature usually rise above, okay, they cannot be easily killed, okay, when they are stabilized by your magnesium chloride. Unlike your rhino are acid labile, okay? So acid labile. So they when you ingest your sipon, okay? So your secretions from the nose, the rhinovirus usually die in the stomach because they are usually acid sensitive. Okay, so the enteroviruses is lighter than rhinovirus. It's 1.34. Unlike your rhino, it's 1.4 in terms of the buoyancy. So this is your enterovirus and you have your rhinoviruses. Now, these are the classifications of your right picornaviruses. We have the entero, okay, enteroviruses, and you have the rhinoviruses. Entero is you have the picornavirus, the poliovirus, the Coxsackie virus. We have the hepatovirus, we have the hepatitis A. But your hepatitis A will not be discussed under your picornaviruses. This will be discussed under your hepatitis viruses, under Dr. Eileen Pasqua next week. Okay, you have the Cobo virus, Ike virus. We will not discuss this because it's not common. We have the Pareco viruses. We will not also discuss. And what we will discuss is your aptoviruses, your foot and mouth. So we will only focus on the enteroviruses, the rhinoviruses, and the aptoviruses, the foot and mouth disease viruses. Okay, we have also cardioviruses. We will not also discuss this. Okay, so these are your enteroviruses. We have the enterovirus A, A71. B, we have the B3 Coxsackie, enterovirus C, which is known as your polio viruses. Okay, and we have the rhinovirus A, B, and C, commonly causing your common colds. We have the hepatoviruses, hepatitis A, as I've said, this will be discussed next week, and we have the foot and mouth disease virus, known as your aptoviruses. Okay, so these are important classification of your picorna viruses. So we have entero, pareco, hepato, apto, and cardio viruses. Okay, so your enteroviruses of human origin are subdivided into 7 species based mainly on the sequence analysis. We have the polio, so we have 3 serotypes of your polio: poliovirus 1, polio 2, polio 3. We have Coxsackie viruses, group A, we have type 1 to 24, but there is no numbers like this. Okay, sandali lang. Okay na dyan? Masasabi lang sa akin is more excuse me for a while. Marvin Zara. Hello. Pasensya na po, may toxic lang akong COVID na pasyente. Okay, so we have polioviruses 1, 2, and 3 serotypes. We have the Coxsackie 1 to 24. Okay, and we have also Coxsackie viruses of group B, okay, type 1 to 6. We have the echo, type 1 to 33, but there are no types, this one. And you have enteroviruses, 68 to 116, but there are no type 72. So these are your enteroviruses. Okay, so these are the differentiating point between your human enteroviruses and your human rhinoviruses. So your polio under your entero, serotypes 1 to 3, Coxsackie 1 to 24, B is 1 to 6, Echo is 1 to 33, and entero is 68 to 116. But there is no, sabi dito, there is no enterovirus 72. Okay, while your human rhinoviruses, there are around 150 okay, serotypes. Okay, so in terms of acid, all of your human enteroviruses are stable, while your rhinoviruses are labile, so they're acid labile. So in terms of buoyancy, okay, so as I've said, your rhinovirus is heavier as compared to your enteroviruses. So the optimal temperature is 37 for your entero, while your human rhino is 33. As I've said, but they are stabilized by your magnesium chloride. So they cannot be easily inactivated, okay, because of the magnesium chloride that stabilizes them. So in terms of the common side, of course, nose, rhino is nose here. Okay, but your throat, all of them can okay, be isolated through your throat. You can do throat culture or throat swabbing for RT-PCR of these okay, viruses. So lower intestine, okay, so since they are acid stable, they can survive in the intestines. Okay, they can pass through the stomach. Unlike your rhino, it's not acid stable. Okay, so your human rhinoviruses okay, include more than 100 okay, antigenic types and fall into human rhinoviruses, okay, human rhinoviruses A, B, and C. Okay, so meaning we can have several episodes of rhinitis because there are around 100 okay, genetic types of your rhinoviruses. So if you had rhinitis in 3 times per year, okay, you can also have again rhinitis in the next year because there are a lot of genetic types of your rhinoviruses. Parang walang immunity. So this is how they replicate. Okay, so your picornaviruses usually enters. Okay, it binds to the plasma membrane, to the host cell. Okay, so on. Okay, so the gene. So after binding, okay, you see the genome. The RNA is released. The single-stranded RNA is released. Okay, so the genome is encoded, uncoated siya. So maghuhubad, okay, tatanggalin yung membrane niya dito, and it will release. Sorry. Hello? Opo. Saan po to? Ay, ma'am, pwede mamaya. May klase lang. May, apay, ma'am. Apay, ma'am. Apay. Sige, text lang, ma'am. Sorry. Okay, so mag-uncoat, this the single-stranded is removed, okay, from the viral RNA, which is dead. So this now, okay, single-stranded RNA will now be translated. Okay, will now be translated into your polypeptide protein. Okay, so polypeptide protein. So after translation, okay, so this will be translated and processed into 1, 2, and 3. So this a single-stranded RNA positive will now undergo okay, conversion. Okay, will now undergo transcription okay, into a negative strand RNA. So from here, okay, it goes here, negative strand RNA. So this negative strand RNA will undergo translation. So the single-stranded RNA will become a negative, okay, negative strand RNA, this one, negative strand RNA. And this negative strand RNA will be replicated, will be translated to become 3, okay, to become 3. Okay, so this negative will now be replicated into a positive RNA. Okay, so maa-amplify siya, dadami siya. Okay, so the single-stranded positive RNA will become and the single-stranded positive RNA will be translated into a negative strand, and this negative strand will be translated into your positive RNA. So there will be amplification of the genes. Okay, so after amplification, they will now form, okay, they will now be translated into your polypeptide protein, okay, and they now combine to form what we call your Picornaviruses. Okay, yung buo na yung Picornaviruses. After the assembly of your Picornaviruses, it is shed off from the cell, okay, shed off from the cell, and it will infect another cell. So remember, it does not take the host membrane, okay, because this is usually naked. Unlike your herpes virus, it has an envelope because it gets the plasma membrane of the host cell, but in here, picornaviruses, it does not. Okay, so which are then copied to produce the positive strands RNA to. Okay, so early in infection, newly synthesized positive translated to produce additional virion protein, this one, and eventually they will be shed off. Okay, so the receptor for polio, okay, so the receptor for polio and human rhinoviruses, okay, are members of the, okay, immunoglobulin generation superfamily, which include antibodies and some cell surface adhesion molecules, like your intracellular adhesion molecules. In contrast, your echoviruses recognize a member of the integrin adhesion, okay, integrin adhesion. Sa polio and rhino, it's usually the intracellular adhesion molecules, yung receptors niya. Okay? While your echo, the receptors are usually the integrin. Okay? Not all rhino or echo use the same cellular receptors. Okay? You have the foot-mouth disease, okay, like your enterovirus 71, Coxsackie 16, causing your hand-foot-mouth disease, uses these receptors. Okay. So you have the enterovirus group. The first enterovirus group is the most important because this is of more of your concern, our concern, okay, and global concern, okay, poliomyelitis. So when we speak of polio, meaning this is an acute myelitis, okay, this is an infection of the spinal cord. So polio is an acute infectious disease that in serious form, it affects the central nervous system. So it particularly affects the motor neuron. So this is the dorsal root ganglion. This is the dorsal part of the spinal cord. This is the anterior part. Your dorsal is your sensory, while your anterior is your motor. So this is where the poliovirus, okay, destroy, particularly the lower motor neuron, the motor neuron. Tinisira niya. Okay, so when the motor neuron, okay, particularly the lower motor neuron is involved, okay, the patient may suffer from flaccid paralysis, lupaypay. So try to elevate the lower extremities, okay, there is sudden drop. So the motor is really 1/5. So that's flaccid. In Tagalog, lupaypay. So however, most polio infections are subclinical. So most of the infections are inapparent. Okay, so these are the properties of your virus. Okay, so they are inactivated when heated at 55, okay, for 30 minutes, but the magnesium prevents this inactivation. So thermally, di ba, magnesium, okay, will prevent them from being inactivated by heat. Okay, purified poliovirus is inactivated by chlorine concentration at 0.1, okay, and higher concentration of chlorine to disinfect sewage. Okay, and polio are not affected by ether or sodium deoxycholate. Okay, so like rhinoviruses, they are also not activated. They are also not affected. So animal susceptibility and growth of barriers, they are restricted to host range and requires primate-specific membrane receptors. Di ba may mga receptors na kailangan para ma-infect ka. So that you become infected, you need a receptors, particularly yung mga intracellular that I have said, mga integrin. So this is the poliovirus. The antigenic serotypes of your polio, we have type 1, type 2, and type 3. So I've said your type 1 is usually the most common, and this is the one causing the epidemic. Okay, epidemic. Okay, so your 2 is your endemic. So meaning endemic, it is inherent nandun na. For example, schistosomiosis is endemic in Leyte. Malaria is endemic in Palawan. Okay, and type 3 is strains that have caused also the epidemic. So 1 and 3 can also cause epidemic, while 2 can cause endemic. Okay, so how do we acquire polio? Okay, poliovirus through its mouth. Okay, from acquisition of contaminated water. Okay, it goes into the stomach. It can survive for around 1 to 3 hours, and it goes into the intestine where they replicate. Okay, so regularly present in the throat and stool before the onset of illness. So before the onset of illness, the patient is now still infectious. Okay, the patient is shedding off the virus in his or her stool, even, okay, even with high antibody levels in the blood. So without infection, okay, without signs and symptoms, but you have ingested this contaminated water, okay, you can shed off the virus for several weeks. Okay, for several weeks, especially if the patient is immunocompromised. Okay, especially immunocompromised, yung mga malnourished child, okay, so they can shed off the virus for a prolonged period of time. So this is how fecal oral transmission, oral ingestion. From oral ingestion, it goes into the Peyer's patches. Okay, so lymph nodes. Okay, so the barrier. From the lymph nodes, it goes into the brain. Okay, it goes into the brain. Okay, brain. Okay, from the brain, it may go, sorry. Okay, oral ingestion goes, it can pass through the blood-brain barrier. Okay, from the lymph node, it can pass through the blood-brain barrier going into the blood, primary viremia. From the blood, it goes into the blood-brain, it passes to the blood-brain barrier. Okay, when it goes into the CSF, the patient may manifest meningitis or encephalitis. Okay, so from the blood-brain barrier, it can penetrate the blood-brain barrier, or it can pass to the, okay, peripheral. So it goes to the peripheral nervous system, okay, from the skeletal muscle, okay, from the here, and going to the axon, okay, axon going to the cell body. So this is an axonal okay, transport, and it can destroy the anterior horn of the spinal cord. Okay, leading to what we call paralytic poliomyelitis. So virus is present in the blood with non-paralytic poliomyelitis. So antibodies appear early, usually before paralysis occurs. So even though if you have already, okay, made up, okay, secreted antibodies, okay, paralysis may still occur. Okay, so antibodies appear early before paralysis. Okay, so we have viremia. So we have the blood. Okay, the virus in the blood, okay, goes into the lymph node. Okay, from the lymph node goes to the peripheral nervous system, from the axon to the central body, going to the CNS. Okay, where it continues to progress along the fibers of the lower motor neuron. Remember, it affects usually, okay, what is affected is usually the lower motor neuron. Now, the lower motor neuron is the one that innervate the skeletal muscle. Okay, so it affects the lower motor neuron, no one to innervate the skeletal muscle. So if no one to innervate, no neuron to innervate the skeletal muscle, then the skeletal muscle will not contract. So if the skeletal muscle will not contract, then the patient may suffer from paralysis. Okay, if it does not contract, the muscle will undergo eventually atrophy. Liliit ng liliit yung muscle ng patient with polio. Okay, so increasingly involve the spinal cord or the brain. If it is spinal cord, it may lead to paralysis. Okay, the brain, it may lead to meningitis and encephalitis. So this is the portal of entry going to the small intestine where they replicate. Then you have the minor viremia. Okay, from the minor viremia, it may go into the spinal cord. Yeah. Okay, never mind this. Okay, sometimes it's usually primary. Okay, so minsan hindi na dumadaan sa secondary viremia. Primary viremia going to the spinal cord and brain and multiply in the neurons. Okay, and lesion mostly is in the anterior horn of the spinal cord. Okay, where it causes flaccid paralysis and it will destroy the neuron. Okay, so leading to residual paralysis. Okay, so this is the receptor, okay, of your polio in the cell CD155 on the cell membrane. This is where they is basically attached. Okay, kung yung SARS-CoV-2 attaches to the ACE2 receptors in our cell, the poliovirus attached to the CD155. Okay, so they do not multiply in the muscle. Remember, your polio does not multiply in the muscle. They multiply in the neuron. Okay, they multiply in the small intestine and they destroy the motor neuron. Okay, they do not replicate in the muscles. Okay, where the point of entry is where they replicate. For example, oral ingestion, they replicate in the intestines. So changes occur in the peripheral nervous system and the voluntary muscle. Okay, so eventually there will be destruction of the nerve cell. So if you destroy the nerve cell, no one to innervate the skeletal muscle and the muscle will not contract. Okay, it is also associated with other pathologic changes. So your poliovirus does not only cause a poliomyelitis or flaccid paralysis, but it can also involve variety. Okay, multi-organ. So the heart, myocarditis, lymph nodes, lymphatic hyperplasia, and it can lead to ulceration of the Bayer patches, and the patient may also suffer from bleeding, like ulceration, like the patient may bleed. Okay, so most of your polioviruses are usually subclinical or inapparent infection. Okay, but mild febrile illness can also lead to severe permanent paralysis. Okay, so the incubation period of polio is usually 7 to 14 days. So from ingestion of this contaminated water, okay, it may manifest like fever, okay, 7 to 14 days, and may range from 3 to 35 days. Okay, so your poliovirus ranging from one, it's mild. So this is the minor illness and recovery in few days. And this is the most common form. So the most common form is usually mild diseases, okay, for your polio. Okay, so other may manifest in poliovirus, we call it your aseptic meningitis. Okay, or otherwise known as non-paralytic poliomyelitis because it affects the brain, okay, brain, not the spinal cord, okay, not the spinal cord. That's why the manifestation is, okay, meningitis and encephalitis. Okay, so when speak of meninges, okay, this is the covering. These are the coverings of the brain, the dura, arachnoid, and pia. So when they are inflamed, the patient may manifest with meningitis and he has stiffness, pain in the back and neck. Okay, so when you were first year, you discuss signs of meningeal irritation like Brudzinski, okay, and your Kernig's. Okay, Kernig's. These are all signs of meningeal irritation, and these are signs of your non-paralytic poliomyelitis. They may last 2 to 10 days, and recovery is rapid and complete. Okay, complete ang recovery. Unlike your paralytic, recovery is impossible. Okay, so this is what we call your aseptic meningitis. So these are signs and symptoms of your non-paralytic poliomyelitis. So if you have the non-paralytic poliomyelitis, we have also the paralytic poliomyelitis. Okay, so this is the predominant complaint. The predominant complaint of your paralytic poliomyelitis is your flaccid paralysis. Lupay-pay siya talaga. Flaccid paralysis. Because what is affected is usually the lower motor neuron. As I've said, this is the one that innervates the skeletal muscle, okay, the lower motor neuron. So there will be in coordination secondary to brainstem involvement. So if the brainstem is involved in coordination and spasm of the involved muscle or the non-paralyzed muscle. So there will be pain, okay, of the uninvolved muscle. And the maximum recovery is 6 months, but there is residual paralysis. So these are some points that can cause also aseptic meningitis. It's not only polio, okay, but also aseptic meningitis can also be caused by your group P Coxsackie virus, your group A Coxsackie virus, particularly the A7, okay, group A7 and group A9. So these are some differential diagnosis of your aseptic meningitis, not only polio. Okay, so this is what we call your paralytic, because this is what we are more concerned of, your paralytic poliomyelitis, because, okay, so we have manifestations of abortive or non-paralytic. Yung abortive or non-paralytic, yun yung aseptic meningitis. Okay, so your paralytic poliomyelitis. So this is an invasion of the motor nerve, which is responsible for, okay, the one contracting the muscle. Now, your paralytic poliomyelitis can be divided into three subtypes. So we have the spinal poliomyelitis, we have the bulbar poliomyelitis. So when we speak of the bulbar poliomyelitis, this involves the brainstem. So if it involves the brainstem, the midbrain, the pons medulla, it is usually fatal because it is here in the brainstem that regulates the heart rate, the respiratory rate. Okay, so if it involves the bulbar, it destroys the neurons in the bulbar and the patient may suffer death. Okay, and we have encephalitic or meningitic, okay, if it involves the brain. So this is a patient with polymyelitis. You see the involved muscle. Okay, so this is flaccid, okay, and able to dorsiflex or plantar flex. Okay, so this is flaccid, and since it does not contract, it will eventually undergo atrophy. Okay, atrophy. Okay, so some patients may also be affected on the upper extremities, but more commonly here in the Philippines is the lower extremity. It's usually unilateral. Okay, but there are cases that is usually bilateral, but more commonly, okay, one side of the spinal cord. So you see one? Uninvolved muscles contraction causing tight cords, pain, spasm. Okay, so we have the paralytic poliomyelitis. Eventually, there will be muscle wasting, muscle atrophy because of the loss of the neuromuscular function. Okay, so this is an example of a patient with polio, okay, myelitis. Okay, so how do we diagnose polio? So throat swab, remember, they usually can be isolated from throat. Okay, so shedding, okay, rectal swab, and there is usually no permanent carriers among immunocompetent. But if the patient for several weeks or until 6 weeks, so long-term excretors in some immunodeficient persons. Okay, so uncommonly recovered. So your virus is uncommonly recovered in the CSF. That's why we call it aseptic meningitis. When you try to culture the CSF fluid for any bacteria, there is no bacteria. Okay, you cannot also isolate the virus. So that's why it's aseptic. It's still sterile, but you have the infection. So cytopathogenic effect can usually occur in 3 to 6 days. So it can destroy the cell in 3 to 6 days. So isolated virus, they have to identify type by neutralization with specific antiserum. They have to know if this is serotype 1, serotype 2, or serotype 3. 1 and 3, as I've said, can cause epidemic. 2 can cause endemic. So more commonly, we use the PCR for diagnosing viral because this is the most feasible, and this is within 24 hours, you can already know the results. Like your SARS-CoV-2, we do PCR. Okay, so only first infection with polio produces strictly type-specific responses. So subsequent infection is usually heterotypic polioviruses. Okay, so immunity is permanent to the virus type causing infection. So if you have immunity, to serotype A, so therefore you have immunity not to type A. So predominantly it's an antibody mediated. So it's not cell mediated immunity. So immunity to polio is usually predominantly antibody. So lower degree of heterotopic resistance induced by infection between type T. So there is low degree of heterotopic resistance to type 2. Okay, so they can be passed from maternal circulation. The antibodies from the mother can pass to the baby, so the baby may still be protected until 6 months of life. Okay, so these are the passive. It only lasts usually 3 to 5 weeks, some passive antibodies, but the virus neutralizing body is the one important. Okay, they form soon after exposure to the virus, often before the onset of illness, and persist for life. Okay, so these are patients who may recover, okay, from this virus infection because they are immunocompetent. So they develop antibodies to fight this. And these antibodies may persist for life. But what we are concerned, if the patient is usually malnourished, okay, immunocompromised, they may not recover from this and they may lead to permanent paralysis. Okay, so in developed countries, commonly infected are the adolescent, while in developing countries like the Philippines, commonly infected are under 5 years of age. Okay, so what we call infantile paralysis. Now, this is brought about by poor sanitation and crowding. Okay, so contaminated and commonly in Africa and Asia. See, okay, Philippines, India, okay, are one of the leading causes ng dumami yung polio before. So we have eradicated, in the Philippines, we have eradicated polio since 2000. Okay, since 2000. Okay, there was a report on the 16th of September 2019, di ba? Nawala ang polio virus since 2000. Okay, 2000, okay, to 2000, okay, August 2019, there was no report. Okay, so the Philippines is already poliovirus free. Until 16th of September, there was a 3-year-old girl from Lanao del Sur, okay, who presented with an acute flaccid paralysis. Pumunta sa LGU, okay, the patient already paralyzed. Okay, so they get the environmental sample, okay, in the Lanao and they found the virus. They also got environmental sample in Manila and waterways in Davao were confirmed to contain also the viruses. Okay? So, nabulabog ang Pilipinas. So, the Philippines, the Department of Health, okay? So, you know what? There was here a case because of the issue there before of Yodeng-Baksha. So, the parents were then afraid of vaccinating their children because of the issues, hot issues of the Baksha before, because of many have died. So they were then adamant to bring their children, okay, to vaccination facilities. So what did the Philippines? The Department of Health said that they have to vaccinate 95% of children under 5 years of age, okay, in order to protect this child from polio. So we have stopped the polio before. Together we can stop this terrible disease again. So this is of concern, like the SARS-CoV-2. So this is a reportable. So between July 1, okay, and August 27, okay, environmental sample tested positive. Okay, so there was a polio virus, okay, so water sample in Tondo. Okay, and they have identified this as your vaccine-derived poliovirus 1. So vaccine-derived. Okay, so this has been excreted by a patient with vaccine. Okay, vaccinated patient. Kaya it's vaccine-derived poliovirus 1. So between August 13 and 22, okay, environmental sample tested for vaccine-derived poliovirus 2 in Tondo and Davao. So they have tested waters in Tondo and Davao, okay, that there is a vaccine-derived poliovirus 2, and they found out that this was genetically linked on 13th of September 2019. Okay, what happened to patient, 3-year-old girl, okay, who presented with an acute flaccid paralysis in Lanao del Sur. So there was a contamination also waters in Tondo and Davao. Okay, so there was a wide, so genetically parehas. So what then? Okay, so the vaccine-derived poliovirus 2 was classified as C. C is circulating vaccine-derived poliovirus 2. Okay, so that was identified now on the 19th of September, and there was an outbreak in Lanao. Okay, Lanao, because pag isang case, kahit isang case lang, even only one case in Lanao, is considered as an outbreak na yon. Okay, for several years, okay, walang case ng polio. So this is circulating vaccine-derived poliovirus 2. Okay, so in Manila, okay, 30 of January 2020, since the emergence of the polio in the Philippines announced by the Department of Health in September 2019, okay, with synchronized polio vaccination campaign rolled out from November to December 2019, okay, di ba, September nangyari, November to December 2019, they have vaccinated almost, okay, 1.5, almost 1.4 million, more than 1.4 million below 5 years old in the National Capital Region and almost 3 million in children in Mindanao. So they were then protected because 95% in Mindanao has been vaccinated. So they have achieved here the herd immunity in Mindanao. Okay, so the call to vaccinate further intensifies. So it further intensifies with these cases recorded in January of 2020, okay, bringing up a total dumami of 16 across the country. Okay, dati isa lang. Okay, and there were already 16 across the country. So all new cases are children below 5 years of age. Okay, so most of the cases from Maguindanao and Sultan Kudarat in Mindanao. Okay, and there was also reported cases in the National Capital Region in Quezon City. Okay, so that was 2019. And 2020, February, up to present, there were no already reported cases of polio as far as I'm concerned because of the wide vaccination. So the transmission of polio is spread through the fecal oral, okay, can only be stopped if under 5 years old are vaccinated. 95% of the targeted children has been vaccinated. So this is must be vaccinated during an outbreak. Kahit na nabakunahan yung ibang pasyente, okay, yung ibang child, they need still to vaccinate again. Okay, so the Philippines is affected, okay, by your circulating vaccine-derived poliovirus 1 and the circulating vaccine poliovirus type 2. So CDB is considered a public health emergency of international concern. So parang yung mga variant na may public health concern ng SARS-CoV-2. So to ensure protection, okay, so there must be vaccination multiple times. Okay, wash hands with soap and water, use toilet, consume food, fully cook, and drink safe water. Okay, so these are the actions done by the Department of Health. So this is Duque, okay, administering, okay, the oral polio vaccine. Okay, so what's the schedule of your oral polio vaccine? So around 1.5 months, first dose, second 2.5, and third dose 3.5. Okay, and during the third dose, you can also give the inactivated polio vaccine, okay, through injection intramuscular. Okay, at 3.5. So pwede pang siyang pagsabayin. Okay, so this was also a concern. Okay, for example, you going to, okay, your parents or you going to countries like America and you are from Philippines, you were then required way back in 2020, okay, that you were then required to be vaccinated with inactivated polio vaccine because you came from the Philippines and they have assumed, okay, na baka ikaw ay carrier ng polio. So they have to be vaccinated. Okay, inactivated polio vaccine na dun sa atin. Okay, so these are the 3 doses of your oral polio and this is your inactivated polio vaccine, only 1 dose. So ang polio nag-mutate din sila. Okay, so both live and killed virus are available. So the formalized sock is prepared from virus grown in monkey. So the killed virus induces humoral immunity, but induce no local intestinal immunity. So yung killed, okay, does not produce IgA, while your live or inactivated produces IgA, okay, in the intestines. So we have the live attenuated virus. So this is the oral polio virus. Okay, so this is live attenuated virus. So they are stabilized by magnesium chloride so that they will not be inactivated by heat, and they are kept at 4 degrees centigrade. Malamig because they are live. So you have to preserve them. Live weakened. So the live polio vaccine infects, okay, multiplies, okay, multiplies, the antibody multiplies and immunizes the individual. It produces IgM and IgG as well as IgA in the intestine, which then become resistant to reinfection. So that when you ingest this contaminated water, you have the IgA antibody against this polio, and it will easily kill the polio in the intestines. Okay, unlike your killed virus, it does not produce IgA. Okay, so the killed and the live virus vaccine induce antibodies and protect the CNS from subsequent invasion by the wild virus. So the gut develops far greater degree of resistance after administration. Yan, live virus. So the GIT, okay, has better, okay, coverage to intestinal immunity. Okay, so type 2 and 3 mutate in the course of their multiplication in vaccinated children. Okay, so the oral polio is used in global eradication. So the live virus vaccine is contraindicated in immunocompromised groups. Okay, in immunocompromised. So you do not give live to immunocom because they cannot produce antibodies. So this live attenuated may, instead of protecting, it may lead to more severe disease. So pwedeng ma-convert yung live into yung live virus into more pathogenic form. Kasi they are not able to produce antibodies among immunocompromised. So immune globulin is effective only if given shortly before infection, before the onset of the symptoms. So next, we move on to your Coxsackie viruses. So Coxsackie, same, spherical, they are naked, so they have no envelope. Okay, it's a single-stranded, same with Picorna, of course. They multiply in the cytoplasm, resistant to ether and acid, sensitive to drying heat by UV, or survive for a long period in water and sewage. So virus has been recovered from the blood in the early stage of natural infection in human, and virus is also found in the throat, okay, and early in the infection and in the stool for up to 5 to 6 weeks. Parang dengue, no? Polio. So virus distribution is similar to that of other enteroviruses. So the incubation period of your Coxsackie is 2 to 9 days. The distinct disease, they can also range from CNS, di ba kanina yung mga A71, okay, B16, Coxsackie. They can also lead to skin, okay, rashes, vesicular rashes, okay, and also cardiac and respiratory. So it can also involve multiple organs. So different serotypes may be associated with particular outbreak. So these are the diseases caused by your Coxsackie viruses. So number one is we have your herpangina. Okay, so this came out in the board exam, Philippine licensure examination. So herpangina, you can see vesicular rashes, okay, and vesicular lesions on the throat. Okay, there are discrete vesicles. Yeah. Discrete vesicles on the palate, pharynx, tonsils, and tongue. So these are common among group A Coxsackie virus. So we have hand, foot, and mouth disease. So you can see vesicular lesions on the tongue and foot, hand, mouth, okay, and in the hand. So this one, okay, and also on the lower extremities, the foot, the talampakan. Okay, oral and pharyngeal ulceration, vesicular rashes of the palms and soles may spread to the arms and legs, and vesicles heals without scarring. So they are associated with this hand, foot, and mouth, A16, and enterovirus 71, your B1, otherwise known, and enterovirus 71. So A16, B1, and enterovirus 71. All of these can cause your hand, foot, and mouth disease. What about your pleurodina? Okay, or your epidemic myalgia. Maya is pain, algia is pain, maya is muscle. So the patient may experience chest pain. Okay, so they fear fever and stabbing chest pain. So they can be mistaken as MI. So one of your differential is MI. So the chief complaint in children is abdominal pain. So this myalgia, chlorodynia, is caused by your group B Coxsackie viruses. Okay, so your group B can also cause your permanent heart damage. So this is fatal permanent damage at any age. Can also lead to myocarditis, your group B Coxsackie viruses. Okay, so generalized disease of infant. This is extremely serious disease. Your Coxsackie is seriously disease also. Multiple organs are, excuse me. Okay, extremely serious. Multiple organs are affected. So rapidly fatal or recover completely. Severe cases, you see, your myocarditis or pericarditis occur first 8 days of life and may be preceded by diarrhea and anorexia. So may be acquired transplacentally. So mother with hand, foot, and mouth, they can pass this virus to their children and it can be acquired through placenta. So to the child. GIT, the site of replication, but seldom with GI symptoms. So here, how do we recover them? Okay, also throat culture, okay, or PCR, and they can be isolated from the stool during the first few weeks. So it can also lead to aseptic meningitis. Yung sinabi natin kanina, one of your differential, okay, is your Coxsackie. So they can be isolated in the CSF. Okay, it can also lead to hemorrhagic conjunctivitis. Okay, so what is the bacteria or serotype of this hemorrhagic conjunctivitis? So later on, we will discuss. So this one. So your nucleic acid detection, your PCR, is the one also being used because it's sensitive to conventional PCR but are less labor intensive. So it's easily done rather than doing your viral culture. Okay, so rapid and sensitive assay. Your reverse transcription PCR test can all be broadly reactive. So detect many serotypes. So PCR is very helpful. So these are your other tests, neutralizing antibody, your immunofluorescence, okay, and difficult to evaluate unless with isolated specific antigen. Sometimes this is serology. Okay, so this is the scenario way back in 2018 for your hand, foot, and mouth disease. So a total of 78 suspected and confirmed hand, foot, and mouth disease cases were reported nationwide from January to February 3, 2018. So there was 83.0 lower compared to the same period last year, 416 in 2017 and 78% na, ay 78 lang sa 2018. So what are the regions commonly had the high cases of hand, foot, and mouth? So the region 4A, and of course the region 7 also had 13, and also the national capital region 14, okay, because of the crowded din. Okay, and case fatality rate, no one died. Okay, so case fatality rate, so it's usually mild. Okay, so commonly infected are the most common sa male under your 1 to 4 year old, 1 to 4 year old. Okay, so this is a case in the Philippines. So most of the cases in the Philippines are your Coxsackie virus A6, the hand, foot, and mouth. So 5%, 100% of the cases, okay, in the Philippines is usually Coxsackie virus serotype A6. Okay, other enteroviruses, these are nice to know. Okay, so we have ecoviruses, we have the enteric, cytopathogenic, human orphan viruses. So these are just nice to know, part of your picornaviruses. Historical terminal were grouped together because they infect human enteric tract, because they can be recovered from the humans only by inoculation of certain tissue cultures. Okay, so there is much higher rate of recovery of virus from patient with disease that form a healthy individual of same age. So kung healthy, you can recover from this ecoviruses. Antibodies developed during the course of the disease. Okay, like your polio, even not in the course of the disease, okay, no symptoms, you can still have the antibodies. But here in the ecoviruses, you can have antibody during the course of that. So this is isolated from body fluids or tissue manifesting the lesions. So this is what I'm saying. Enterovirus 70 is the one causing your acute hemorrhagic conjunctivitis. Acute hemorrhagic conjunctivitis. So this was enterovirus 70. Okay, so the other one is enterovirus 71. Is the one that causing your meningitis, encephalitis, and paralysis. Now that's why this is one of your differential diagnoses. You have your Coxsackie A17, you have enterovirus 71, poliovirus causing all of these, CNS infections. So these usually are common during the summer outbreak. And epidemic usually in young children of a febrile illness with rash. Yung mga ecoviruses. So serologic tests. So neutralizing and hemagglutination inhibition antibodies are type of specific and many persist for years. So the epidemiology of echo is similar to that of other enteroviruses. So of course, your enteroviruses are brought about by contamination of these several group na yung mga waters, irrigation, groundwater. Saan din papupunta? Makukuha ng shellfish, recreation, water supply crops, and aerosols, and they can be acquired by humans. Oral fecal pa rin. So lastly, you have rhinoviruses. So there are around 150 types, and 70% sequence identified already. So your rhinoviruses, your human rhinoviruses A to D, human rhinoviruses A to D is having a buoyant density of 1.4. So they are unstable at pH, so they are acid labile, sabi ko. And thermostable when they have the magnesium chloride. So these are infectious to humans, gibbons, and chimpanzees. So organ culture of ferret and human tracheal security may be necessary for some fastidious strains. So most grow better at 33 degrees. So it's the nose 33 degrees. So they usually thrive best at the nose because of the temperature. 37 yung enteroviruses. So there are more than 150 serotypes. And new serotypes are based on the absence of cross-reactivity in neutralizing. So human rhinovirus 87 is now considered the same serotype of human enteroviruses. So the portal of entry, high titer of virus in the nasal secretions. 2 to 4 weeks after exposure, titers fall but illness persists. So yung sipon, mamawala yan 7 days. But the effect of the sipon like cough is now usually may persist for 2 to 3 weeks. So bakit hindi nawawala? Yung cough mo, kasi kagagaling mo lang sa sipon, yung sipon mo nawala. But the non-productive cough may persist for 2 to 3 weeks each year. So sa isang taon, ang average di magkasipon is 1 to 2 episodes per year. So these are the symptoms of your rhinitis. So neutralizing antibody to the infective virus developed in serum and secretion of most persons, your IgA. So depending on the test used, estimate of the frequency of response some range from 37 to greater than 90. So the antibody developed 7 to 21 days after the infection. So the antibody persists for years but decrease in titer. Kaya magkakaroon ka na uli ng sipon kasi bumababa, nagbe-wane off yung antibodies natin sa rhinovirus. So they are prevalent in the lower socioeconomic. So they can be acquired through your respiratory secretions, contaminated inanimate object, and they can survive 4 hours in a contaminated surfaces, parang SARS-CoV-2, and infection is highest in infants and children. So how do we treat? We give just antiviral drugs, or usually this is supportive, water and multivitamins. And sometimes they give may mga allergic rhinitis, so mga intranasal corticosteroids. So that ends our lecture for your Picornaviruses. So any questions? Do you have some concern?